Hello everyone. In the last module, we have learned about the cytoskeletons. The filaments of cytoskeletons. In that, we studied microtubules and intermediate filaments. In this module, we are concentrated on actin filaments or microfilaments. Microfilaments are made up of two strands of actin filaments. In that, they consist of actin subunits, and these two microfilaments of the actin subunits get intertwined and they form the filaments called actin filament. This actin filament is very important for the maintaining cell shape as well as the movement of the cells. So, we are going to study about the actin filaments in this module. Learning objectives of these modules are introduction, actin filaments, structure and organization. In that, we are going to see actin polymerization, accessory proteins, actin binding proteins and treadmilling. Cytoplasm shows cytoskeleton in form of microtubules and microfilaments in case of plant cells. Microfilaments seen as thin fiber under electron microscopy with a high voltage electron microscopy used for greater penetration. It appears as microtrabeculae lattice, that is MTL, a 3D structure just below cell membrane. Spaces enclosed in the network are 50 to 100 nanometer in length. These are shown to aid rapid diffusion of fluids in cytosol. The network filaments are in contact with endoplasmic reticulum, microtubules and ribosomes. Mitochondria are free, therefore can move around. Chromophores used or the pigment to study the structure. These show rapid movement like contraction and expansion within few seconds. It was observed that this movement is due to change in structure of MTL. During contraction or at the time of contraction, microtrabeculae shows beaded structure, and during expansion they return to their fixed position. Studied using immunological techniques, microfilament shows presence of contractile proteins. The major component is actin. It is an abundant protein in a cytosol, occurs as globular form or G-actin form with a molecular weight of 42 kilodalton. It can quickly polymerize to other form, showing the straight parallel fibers structures. It is several μm in length and contains thousands of monomers. Cytoplasmic actin can also form 6 nanometer wide double helical array of globular actin molecule. These have affinity to other contractile protein. Myosin, which is bind to give a decontractive form, that is myosin with actin. Actin filaments. Actin is the most common cytoplasmic protein globular actin subunit, or it is called G-actin, is connected to two more actin subunits, one above and one below, with the help of binding site to form filamentous actin, that is F-actin. Actin filaments exhibit polarity plus and minus ends because all the actin monomers are oriented in the same directions. Since each actin monomer is rotated by 166 degree, it has the appearance of double-stranded helix. The figure shows structure of a single G-actin monomer. Assembly and disassembly. G-actin monomers bind to ATP before being incorporated within a filament. Although it is not essential that G-actin is bound to ATP for polymerization to occur, it does not increase the ability of G-actin to incorporate itself into the filament. To begin the assembly of an actin microfilament, 3 G-actin monomers must nucleate to form a trimer. This trimer is called a nucleus. From this starting point, G-actin monomers which have bound to ATP begin to polymerize onto both sides of the nucleus. The G-actin monomers have two ends, a barbed end and a pointed end. Because the G-actin monomers are added onto the F-actin in the same orientation, actin filaments have two distinct ends. The ends are called plus end and minus end. The filament always arranged with the barbed end at the plus end of the filament and the pointed end at the minus end of the filament, which is shown in the figure. If a high concentration of ATP actin subunits are available, there will be rapid polymerization at both the ends of the nucleus. However, as the concentration of ATP actin subunits decreases, the rate of subunit addition at the minus end of the filament is reduced. Because actin is an ATPase, the bound ATP and is hydrolyzed to ADP. Due to these subunits near the minus end of the filament will have bound to ADP. Actin polymerization. Nucleation phase, formation of stable actin nucleus, a complex of 3 actin monomers. Elongation phase, where the removal of the capping protein plus an exposed monomers added at the plus end with the help of elongation factor, for example formin. Steady state phase, the formin binds profilin with the pre-attached G-actin. The concentration of actin monomer in the cytosol will either favor disassembly or assembly of the actin filament, and these values are known as the critical concentration. So, all these explained phases are involved in the polymerization of the actin. When the concentration of free subunit exceeds the critical concentration, the filament elongation occurs spontaneously. Free subunit concentration is higher than critical concentration at the positive end and lower than the critical concentration at the negative end, which have shown in the figure. In the steady state, the plus end grows about 5 to 10 times faster than the minus end. It is for this reason that microfilaments are described as assembling from the plus end and disassembling from the minus end, because subunits are added to the plus end whilst being removed from the minus end. The filament position is continuously moving. This movement is called treadmilling. Association of other proteins with the actin in microfilament were shown in the following diagrams. Myosin have distinct head and tail structure, The head binds and brings about hydrolysis of ATP. The tail differs depending on the type of myosin. Myosin of a few types, right, type 1 and type 5, act as a motor protein that move along the actin filaments. The head attaches to the actin filaments and tail to the cargo. By using energy from ATP hydrolysis, it moves toward the plus end of the actin filament like it is walking on the two legs. We can see in the figure an actin, that is the thin filament, and the myosin, which is the meromyosin. Actin filament polymerization take place in 3 phases: nucleation phase, elongation phase and steady phase. During the nucleation phase, 3 actin monomers form a stable complex called actin nucleus. The removal of capping protein is necessary for the filament elongation to begin. During the elongation phase, monomer addition takes place at the barbed or the plus end of the filament, which is facilitated by certain elongation factors such as formin. Formin then binds to porphyrin with the pre-attached G-actin. Porphyrin maintains a constant influx of actin monomers by promoting ADP to ATP nucleotide exchange on G-actin. Thus, ATP G-actin monomers are added to the growing actin filament. Accessory proteins. In general, accessory proteins serve some important functions like stabilizing and destabilizing microtubules and actin filaments, nucleation, polymerization, their positioning into the cell cytoplasm, interaction of the cytoskeleton component with the other proteins. Microtubules, associated proteins, that is MAP, MAPS and actin binding protein ABPS. We have already seen about MAPS. Now, in this actin binding proteins ABPs, since most of the ABPs bind to the same loci of the actin surface, they either compete with the another negative operability or form ternary complex or a positive cooperability. The actin filaments mostly interact with microtubules and or intermediate filaments via linker proteins. There are various types of ABPs with their major roles being disassembly and assembly. ADF or Coughlin family. Actin depolymerization factor ADF or the Coughlin family of proteins is present in almost all eukaryotic cells. They are smaller and have different isoforms. They recycle actin monomer involved in cell division and membrane truffling. Porphyrin family. This family of ABPs is one of the highly expressed cytoplasmic protein. These are known to be high affinity monomer binding proteins and are involved in the exchange of actin monomer nucleotides. Gelsolin superfamily. These are found in all eukaryotes. Example of the family include severin, valine, CAPG, adservin and gelsolin. These proteins are involved in serving and capping of actin monomers as they swim in the plasma. This ultimately results in their sequestration by vitamin T binding protein, that is DBP, and subsequent removal in the liver. The small proteins belonging to this class were first discovered from the thymus gland and hence are called so. These are widely distributed and highly conserved. T beta 4 is composed of amino acid which possesses hydrophobic residues to a very lesser extent, indicating that it cannot have a well defined structure in the solution. 5. Capping proteins. Capping proteins bind firmly barbed end of actin filaments which prevents exchange of actin monomers. They also play a role of actin polymerization by stabilizing actin monomer and oligomers, which is essential for nucleation. Examples are CAPZ, that is barb end capping protein, and tropomodulin, that is pointed end capping protein. In slide 12, we can see the actin binding proteins. We can see the different actin binding proteins in this figure. How is growth of actin filament is regulated, which is represented in the in this flowchart diagram. In this, binding to the inositides inhibits pointed end depolymerization by ADF and coffilin, actin monomer sequestration by profilin, and the capping of barbed end filament end by gelsolin and capping protein. Inactivation of coffilin reduces pointed end depolymerization. Inhibition of profilin releases ATP bound actin monomer, which therefore become available for barbed end polymerization. Inhibition of gelsolin and capping protein liberates free barbed ends, which allows filaments to elongate. So, the binding of phosphoinositides to these actin binding proteins favor the growth of actin filaments. To summarize this module, we have studied about structure and organization of actin filament, which is the smallest filament of all, having 7 nanometer diameter of the filament. Actin filaments are important for the maintaining of cell shape, and it is also important for the movement of the cell. We have also learnt about the polymerization of actin and control of the growth of actin filaments, or we can say the regulation of growth of actin filament. In the next module, we are going to learn about the functions of all the filaments that we have studied in cytoskeleton. Thank you.